She is also the Board of Research Studies member in Periyar University, Annamalai University, and Madras University, and also a member of UGC Expert Committee to select women scientists for postdoctoral fellowships. She has completed 14 research projects funded by DBT, ICMR, UGC, and DST, and has acquired patents for a novel drug against hepatocellular carcinoma and diagnostic biomarkers for oral submucous fibrosis. She has authored 4 chapters in books and has around 205 publications in various reputed international and national journals, and has submitted 17 sequences in GenBank repository. These are clear evidences of her dedication and commitment towards academic and scientific excellence. Her tireless efforts and tremendous contributions towards scientific research brought her appreciations and laurels across the scientific community, and she has received numerous national and international fellowships and awards. She was conferred with the DBT Overseas Associateship Award for collaborative research on mucin gene expression in eukaryotes, Hershey Medical Center, Penn State University, USA. She was the recipient of INSA Royal Society of London Award for collaborative research on Helicobacter pylori post-interactions at University of Leicester, UK. Academic Achievement Award from University of Madras, UGC BSR Distinguished Scientist in 2011, UGC BSR Faculty Fellowship in 2015, M.K. Nambiar Memorial Oration Award in 2013 from Indian Association of Biomedical Scientists. Absolutely, her contributions and achievements to the field of research are exemplary and a milestone. So with this brief introduction, I now invite Dr. Niranjali Devraj to take over this session. Over to you, ma'am.
Sorry, I had my microphone turned off. Okay, thank you very much for this introduction. And I think we'll start right away before we have, further internet connectivity problems. So that just 1 minute, I'll get this, get the presentation done. Okay, can you see the slides? Any one of you there?
Yes, ma'am.
My mic. Okay, so I start now. So this is a topic that I had given, treatments for emerging infections by targeting host cellular entry pathway. So when we say of pathogens into, say, the deeper tissues is through the mucosal surfaces, which are present in the respiratory tract or in the gastrointestinal tract or in the urogenital tracts. But whatever, you may come into contact with a pathogen, but that is not enough for an infection. The pathogen needs to adhere to these mucosal surfaces. So adherence is actually the first initial physical interaction of a microbe. It may be a pathogen, it may not be a pathogen also, with a host cell. And it is a key first step which determines what will happen next, whether a disease is going to be caused or not. So what will we do? Like you think the host, that is in this case the human, is going to just sit as a silent spectator and allow the microbe have its own way? No. Now, there are a number of defense mechanisms which will be working day and night at these cell surfaces to prevent these intruders. But these surfaces are also continuously being bathed by secretions containing enzymes and antibodies, and organism can be swept away. But at the same time, the pathogens can still get a foothold by taking advantage of weak host defenses. Like if our defense is not good, then the pathogen can get a foothold. And the initial interaction of a microbe is obviously at the surface. And that is a logical place to start if you want to prevent an infection. Because after a microbe adheres to a cell surface and then, gets internalized, or if it is going to secrete its toxins like the Vibrio cholerae into the cell, then, you may have to use antibiotics, you may have to use, several other drugs to either kill the microbe. But in this case, if you're going to block at the initial step itself, the initial adherence step itself, that would definitely be the logical place to start with. And that will be much easier also to control the infection. So as you can see in this slide, please tell me if the slide doesn't move, right? Because sometimes it happens.
Yes, ma'am, it's moving, ma'am.
So as I said, there are these normal cleansing mechanisms, like there can be airflow, there can be fluid flow, like secretions, the ciliary action of the respiratory tract. Then you also have peristalsis in the intestines, desquamation, that is shedding of the cells. Even though the microbe may adhere to the cell surface, the cells can be shed, and therefore the microbe will not be able to cause an infection. It can be excretion, or sometimes even coughing can help not help the microbe, help the host, so that the microbe doesn't get into the host epithelial cells. Sneezing also. But in the case of endothelial surfaces, it can be even the blood flow. So you may have microbes, they can be viruses, they can be bacteria, whatever they are. In an uncolonized mucosa, you can see they're just washed through. But in a colonized mucosa, the bacterium or the pathogen has somehow, been able to breach this defense system of the host and adhere to the host cell surfaces or to the mucosa and start colonizing there. And then it becomes difficult to eradicate the organism. So there's another picture to just show, there's like a cartoon where you see that adhesion, like you can see here, there is adhesion here. When there is adhesion, then what happens is, even if you're going to use some cleansing mechanisms, it will be difficult to remove these organisms. But if you're going to prevent the adhesion itself, then you can easily wash out the organisms. So that's just a simple cartoon. So in this cartoon, it looks very simple. It's a highly simplified picture that we are seeing here, but actually speaking, it's not all that easy, because you don't know to what surfaces or surface molecules that this organism is adhering to. And unless you know that, you will not be able to inhibit that particular adhesion, because not all pathogens are going to adhere to a cell surface in in an identical manner. So therefore, each pathogen has its own receptors or binding sites on the cell surface, and that has to be identified if you want to block that particular initial adherence step. Now, most of these studies that have been done so far, they have been performed in vitro. That is, you have your surface molecule that you think will adhere to the bacterium, and on a, say, on a immobilized on a plate, or a 96 well plate, or on a TLC plate, or on a cellulose membrane on some surface, and then you allow the bacterium to overlay on this particular surface. So these are what are known as in vitro studies. Now, in vitro studies have an advantage because they provide ready assessment of the various conditions of incubation. Like you can incubate at, say, 37 degrees Celsius at neutral pH, pH 7, etc. And also you can use defined substances for the adherence process. Like you can use a peptide, or you can use a glycopeptide, or whatever it is, as the surface, if you feel that is the site or binding site for the pathogen. But it also, these simple methods also have their own disadvantages, and because you're not talking about the physiological, or you're not taking the physiological defenses into consideration here, which is there in an intact tissue surface. So that is being bypassed, and you might be over interpreting the data that you are getting, and you might think, oh, I have actually identified a particular binding site, which may not be true. So you have to be very cautious when you do such in vitro studies. Yes? Any problem there with the mic, or
Participants,
no?
please unmute your mics.
Okay. So nevertheless, in vitro methods can provide useful data, and we have to make use of these methods also, how simple they may be. So you can see in this particular slide, this is one.
Ma'am? Ma'am?
Also, you have turned it off, I think, right? Is it all right, Uma, or anybody?
Yes, ma'am. Yes, ma'am. You are
You
audible,
there?
ma'am.
And I think somebody turned off my mic also, right? Microphone.
I'll see to it,
Okay.
ma'am.
So, Enema?
Yes, ma'am. You please continue, ma'am. Your voice is
Yeah,
audible.
I think yeah. Okay. Okay. No, not that the microphone was turned off, but I didn't do it. It was done by somebody else. Okay. By mistake, maybe. Okay.
Okay, ma'am.
So let's go to the. Let's go to the slide. So here you can see Shigella. Why do I call this my very favorite organism? Because I've been doing, I mean, not me alone. My research group has been doing extensive research in this particular organism, that is the Shigella dysenteriae, and we have got some good publications also out of the work we have done here. So this particular organism also, you can see how it adheres, and it's just a picture to show how it adheres. And then this is all the work that has been done, all the signaling pathways, etc., to finally show how Shigella causes this infection. So the initial interaction is with a toll-like receptor, and so on all these signaling pathways. And finally, it also, in a way, this infection can protect, cannot enter into an adjacent cell, because a protective, mucus is secreted by the adjacent cell. But then, if there is a breach in this, then obviously in this production of this mucus, then obviously the Shigella can go on infecting adjacent cells also. So after doing all this work, mainly based on the initial adherence. Now coming to another organism, which is also one of my favorites, and that is the Helicobacter pylori. A person can have 6 packs, 8 packs, whatever it may be, but if the person is going to be infected with this organism, and that is Helicobacter pylori, you can see it, swimming in the gastric antrum there. And if the person is going to be infected with this organism, then it's that is the Helicobacter pylori, then he is going to end up getting ulcer, initially gastritis, then ulcer. Finally, even if there is a chronic infection, it might even lead to gastric cancer. So when you look at the world, the population, half of the world's population, especially those in developing countries, they are thought to be infected with Helicobacter pylori. All of us probably have been infected, or we have a Helicobacter pylori infection. And this chronic infection, as I said, can lead to gastritis, peptic ulcer, and finally to gastric cancer also if it is untreated. So that shows, and you can also see how the bacterium, that is your Helicobacter pylori, adheres to the cell surface, and it has specific structures. Now here, these are carbohydrate structures, oligosaccharide structures, to which the organism adheres, and after that adherence, it secretes its toxins like the Ka and the Vac into the epithelial cell. But like before this, that is, if there is no adherence, this secretion does not take place, and the organism doesn't actually invade the epithelial cells or invade the mucosa. It just sits on the surface, and that is the initial point of not entry of the organism, but the entry of its toxins to cause all these symptoms that we had already seen. So what is so special about this cell surface is what we have to see now. As we see here, if you look at the cell surface, you have what is known as the Glycocalyx. It's like the icing on a cake. Okay, so you have, and mostly these are sugars. They may be glycoproteins or glycolipids. Otherwise, collectively they're called as glycoconjugates, and they give rise to the Glycocalyx. And this is a surface structure to which several organisms, they may be viruses, other cells, and bacteria can adhere to different structures on this Glycocalyx and cause a disease. So way back in the 1950s itself, this person, Beachy, actually, he's the one who had done so much of work, or he's the one who initiated all these studies on the initial adherence of bacteria and pathogens, and how it is important for the pathogenesis of that particular organism. So in most cases, we have to focus on the host and not the bacterium, because you cannot manipulate the bacterium. You have to manipulate the host. And how do you do that? You can't manipulate the host also, but then you can use some drugs, etc. When I say drugs, they're not antibiotic. I'm not talking about the antibiotics which are given for normal bacterial infections, but these are known as mimetics. They can be peptide mimetics, or they can be glycomimetics, which are used to prevent this initial step, which is known as the adherence. So it again, on a slide like this, it looks very, very simple. You have the bacterium, and you have these structures which protrude from the surface of the bacterium, and they're known as fimbriae. And these proteins, or the fimbriae proteins, can bind to this cell surface molecules. Here we are looking at carbohydrates, we say, because in most cases the cell surface molecules are carbohydrate in nature or sugars in nature. So therefore, the bacterium, when a protein binds to a carbohydrate, we call it as a lectin. So the bacterium has a lectin and the carbohydrate is on the cell surface, the surface carbohydrates. Now, if this happens, then a disease can be caused. But if you can use glycomimetics, that is, if you know the structure of this carbohydrate to which this lectin is specifically binding, then you can actually, synthesize such glycomimetics and use them, and the glycomimetic will go and bind to the lectin and prevent the actual receptor binding to the lectin on the bacterial surface, and the bacteria will be able to bind to the cell surface. In the second, an alternative method, maybe you can use a lectin drug. That means something which is similar to the bacterial lectin. If you are going to introduce that into the cell, then what happens is this lectin will instead go and bind to the cell surface receptor and prevent the bacterium from binding. So in both ways, you're preventing the bacterium from binding to the host. In that way, you're actually focusing your attention on the host. And, many such experiments done, and drugs have been identified which can block this initial adherence for some disease conditions. I'll show you a slide for that. Okay. So as I said, such how do you know what this is, what this carbohydrate is or what this protein is? So you should be able to know what it is. Like, you can't synthesize an entire protein, or you cannot synthesize a large carbohydrate molecule. It's very difficult. All that you can do is, if you can identify just maybe a tetrapeptide in a protein, or a tetrasaccharide, or if sometimes even a disaccharide in a carbohydrate, which will be the specific receptor for that bacterial protein, then you can use that to block this adherence. So there are some, early days, we had used simple techniques, because those were the ones which were available at that time. And you can see here, you can use this simple method for studying bacteria host binding sites, and they are known as overlay techniques. So all that you have to do is you immobilize a part of this sugar, right, or part of this carbohydrate, which you think is going to be the receptor. That's why I put it in, inverted commas there. What you think may be the receptor, immobilize it on, you can do it on a TLC plate, or you can do it on a if it is a TLC plate, it has to be a glycolipid, because that is the one which can bind to the TLC plate. If it is going to be a protein, then you have to use a SDS page and then transfer it to a nitrocellular membrane, which is cellulose membrane, which is just like a Western blotting technique. And you can also use plastic wells, and these like the microtiter plate wells, where the wells can be coated with this particular receptor. And after that, you add your bacterial suspension. That is the meaning of overlay here. But before that, you have to block down specific sites. You can use another protein, like a common protein like BSA, or some detergents like Tween 20 or Triton X100, and bacteria can just go and stick to any surface. But just sticking to a surface doesn't mean it is binding to a particular binding site. So we are going to block all these other non-specific sites to which the bacterium can bind, and then allow only these specific sites to be exposed so that the bacterium can specifically bind there. And after that, you wash off whatever, because not all, now you add 10 to the power 5 or 10 to the power 7 CFU of the bacteria. That doesn't mean all of that is going to bind to the receptor. So whatever is not bound, you just wash it off, and then you can do an autogeography. In this case, you can see they have actually run the glycolipids on a TLC plate, and then overlaid this, so the glycolipids can get separated, and then do a bacterial overlay. Now, when you do a bacterial overlay, what is surprising here is, now if you look at the thickness of these bands, which tells you about the concentration of these, individual glycolipids, you can find that, the one which has a minimum concentration is the one to which the bacterium is binding, specifically. So that shows that, it's not as if you have an abundant amount of a glycolipid, that is where the bacterium will bind. It might even bind to a very minor glycolipid, and which may be the actual receptor for that bacteria. Then you can do it on as I said, on an STS page, where you run an STS page and therefore separate the proteins, and you want to know to which specific protein the bacterium is binding. So you overlay with the bacterial suspension, and you can see here, I mean, normally when you do an experiment, you don't get such clean bands where you just find one bacterial protein band binding to this. But, this is just a picture that has been taken from somewhere. But when we did our research, we never got such clean blobs, So when you do an overlay, so this is just to show how this binds to a specific protein, which is, about maybe 50 kilodalton, less than 66, and, more than 45. So that is the specific binding that you see. Now, this reminds us of another technique that everyone uses today, the arrays, the microarrays. Just like microarrays, DNA microarrays, you have even, you also have glycoarrays. And the glycoarrays are very similar. The technique is actually, the technology that you use is actually very similar to this bacterial overlay. So it is from these older methods that they have actually improvised and come to something called a microarray. So this is the microarray. You can see now on the surface, cell surface, you have several different carbohydrates. You can isolate those carbohydrates, or you can, if they're with smaller molecules, you can even synthesize them and then print them on a chip, a glass plate, you say, a chip. And, like say this one here is what you may be seeing somewhere here, right? So you actually print it on the surface. So you have different oligosaccharides, bound to the whatever that surface in which you have immobilized these carbohydrates, and then you allow the protein. So you have a protein suspension, but you don't know which protein or peptide is going to bind to which oligosaccharide here. So just again, it's like an overlay, and you can see out of different proteins, this protein is the one which binds to this specific oligosaccharide. And if you use a fluorescent label or something like that, you will be able to identify the binding site. So as I said, it's almost the same as that you had seen, which was done years back when I was doing my research. Okay, so these are known as Glycan microarrays for the analysis of glycan binding proteins, because as I said, the cell surfaces, most of the cell surfaces have these glycans or sugars protruding out of the cell surface, and that is the obvious site for pathogens to come and bind. But there must be some complementarity between the pathogen's ligand and the receptor on the cell surface. It's something very similar to an enzyme binding to a substrate very specifically. That specificity is what is important. So why are people not using this glycoarray technology very often? The reason is that the limited availability of, or you need a different carbohydrate structures. So if you isolate a complex carbohydrate, then you have to use endoenzymes which will break that carbohydrate to smaller oligosaccharides, and then immobilize them on chips like this, and then overlay with a bacterial suspension or a protein suspension or whatever it may be. But or you have to synthesize these structures, and the cost of synthesizing these structures is going to be very high. And if you can get them naturally, like if it's a natural carbohydrate which can be used, it will be good. But you don't have many such structures which are available naturally. So this is the Glycoarray is actually a new type of analytical chip that can rapidly analyze. So you don't have to use one by one. You take an oligosaccharide, remove all this oval, then find that the binding is taking place. Instead, you can have an array to which your protein can be added to see whether it is binding, so that you can identify the binding site. Okay, so there are Glycan libraries, like you have DNA libraries, etc. You have a Glycan library also. So for that, you can have all the possible synthetic glycans that could be synthesized. But as I said, it's going to be very, very difficult. It's almost impossible. And it's going to be very costly also. Or you can isolate these glycans from natural sources. So, but when you do it from natural sources, what is important is the purity of the glycan. So, like the steps, you may have to, do many steps to actually get a pure form of this glycan, so that you have a library of all the possible glycans that may bind to a particular protein. Okay, and then when you want to immobilize it on a surface, obviously that glycan, there should be a functional group that will allow the glycan to be immobilized on a glass surface or whatever the compatible surface is. So that derivatization of the glycan is also important. So some of the so there are several limitations, but some Glycan libraries have been assembled, and from these, a lot of data has been generated, and a lot of information has been generated using these Glycan libraries. So what are the sources of natural glycans? Because they are much easier than getting them is much easier than synthesizing these glycans. So you can use milk oligosaccharides, those that are present in milk, and you can see all these oligosaccharide, disaccharides are present in milk, which can be isolated. Or you can use a complex carbohydrate and break it down with the help of endoglycosidases. Like you can see here, it's breaking down a beta-1,4 linkage. And wherever you have these linkages, this endoglycosidase is going to break it down so that you will get smaller oligosaccharides. Or you can even isolate these glycans from plants or from bacteria. But whatever you may be doing, you should have very good purification checks, and you must be able to test the purity of the glycans that you have obtained. So what are the applications of these Glycan arrays? They can be used to identify protein and also antibody interactions with various glycans, and bacteria interacting with these glycans, or viruses interacting with these glycans, and even cells, cell-to-cell interaction also, as you can see here, the cell-to-cell interaction also can be identified. Now, using these glycoarrays, and using the conventional or the classical technique that I mentioned earlier, the overlay technique, actually we were able to identify in patients with cystic fibrosis, and which is aggravated by Pseudomonas aeruginosa. It's not it's not Pseudomonas aeruginosa that causes disease, because this is a genetic defect. But then this aggravates the disease, and you find that a particular disaccharide, the gal beta-1,4 GalNAc, was identified as the receptor for this Pseudomonas aeruginosa. You can use this as a glycomimetic to prevent the binding of the Pseudomonas aeruginosa to the mucus that is present in the cystic fibrosis specifically respiratory mucus, and that has actually reduced I wouldn't say that it cures the disease, because that is a genetic defect and you cannot cure it with all these drugs, but it actually reduces the symptoms. Like these children have difficulty in breathing, digestion, so many different problems, and this could be able actually reduce the symptoms, and that is why it is used. And again, with glycoarray techniques for H. pylori ulcer, the disaccharide sialic acid with lactose, for otitis media, which is caused by Haemophilus influenzae, for kidney failure caused by E. coli toxin. For all these, they have found out the receptors that could be used as glycomimetics using these array techniques. Okay, now all this time we have been looking at the glycocalyx, or carbohydrates on the surfaces of cells. But then it is not always the carbohydrates that are good for all organisms. It's not the carbohydrates or the glycocalyx on the surface which is going to act as a receptor. It is specific for some, maybe for most organisms. But there are other molecules also which are present on the host cell, and they are known as lipid rafts. And what people say is that there can be no better place to gain control of the host cell than through the initial contact with the lipid raft. So that means the organism is actually contacting the lipid raft. So these are lipid molecules and not just carbohydrate molecules, which are found in the plasma membrane. And if you can, so what are these lipid rafts? As you can see in this picture here, they're actually dynamic structures, but they're very thick and stable structures. Their size is, in the nanoscale, that is nanometer scale, that is about 10 to 200 nanometers, and they are found in regions of the plasma membrane. So in the plasma membrane, you will have regions which are raft regions, and you also have regions which are non-lipid raft regions. Okay, so that's why, people say, we have been talking about the the fluid mosaic model of the plasma membrane, and this is a more recent model of the plasma membrane, where you actually, the mosaic part is more important. The mosaic part is because you have patchy areas, right? Like one patch will be everything clustered together, lipids and proteins clustered together, forming something like a raft. Okay, whereas you will have other areas in the plasma membranes which are not clustered like the raft regions. So that is why you call this as a mosaic within the fluid membrane that is present in the plasma membrane. So if you look at the lipid rafts, it's enriched with cholesterol. You have glycolipids. You can have phospholipids, and you also have this Gpi anchored proteins. That is a glycophosphatidylate inositol anchored protein. And this is GPI is actually a post-translational modification of the protein, where a glycolipid is added to the protein. And this GPI anchor is very important in the case of microorganisms that bind to the cell surfaces. So you have, what are the functions of these GPI anchored proteins? They are actually involved in membrane transportation, protein transportation, cell adhesion, and cell wall synthesis, and also in cell surface protection. So in humans, there are something like 150 different GPI anchored proteins, and they may have various roles. Like it may act as a receptor, it may act as an adhesion molecule, it may be involved in transcytotic receptors, transporters, etc. So now, in this lipid wraps, you have, it's not as if all of them have the same lipid formula, the same protein formula. It's not that way. There is a lot of diversity there. So different organisms can bind to different regions of the lipid rafts, for which they have a specific complementary sequence. And if you look at the structure of these lipid rafts, they're like solid bits which are immersed into the fluid environment of the plasma membrane. So those are the lipid rafts. And functionally, what are these lipid rafts doing? They may be involved in plasma membrane signaling, because many of the signaling molecules are actually present in these lipid rafts, as well as exocytosis and endocytosis. When an organism binds to these lipid rafts, then they can be endocytosed. And after, they have gone through a particular life cycle, then they can be ejected out of the cell by exocytosis. But all this happens within these lipid wraps. So if you look at it from pathogens point of view, such lipid wraps which are found on the membrane of the whole cell is like, you have a raft in the middle of an ocean. So the ocean there is the fluid membrane, and you have some which is floating there, and that object is like the raft. So there are some proteins which protrude out of this, like you can see here, protrude out of these lipid rafts, and they are binding sites for microorganisms or for pathogens. And so in lipid wraps, you can see there can be a binding step from to the protruding proteins. Then the microorganism can enter into the cell, and it can get assembled into a full virion or whatever it is, and then it can exit. That's what I said. It can be endocytosed and also exocytosed from these lipid wraps. And some microorganisms, for example, even H. pylori, it exploits the cholesterol that is present in the lipid wraps. And sometimes these microorganisms not only bind to these lipid wraps, but they also can modify the lipid wraps in a way so that the microorganism will be able to survive there. Like you say, dwelling means they're going to remain in the lipid raft itself, and they're not going to be internalized at all. And within the lipid labs, so their entire life cycle can take place within the replication, and all the entire life cycle of these microorganisms can take place in the lipid rafts itself. So why am I talking about these lipid rafts, and what are the significance? That is what's important here. Specifically, as I told you, binding is the first step, and people talk about the SARS-CoV-2. Everyone talks about it these days. And even though you cannot do much research, not everyone will be able to do research on this organism. But then we should be able to understand the mechanisms by which it is causing the disease, which itself is largely unknown. So in this case, what happens is, you can see the SARS-CoV-2, it is docking onto a lipid raft. Actually, the ACE2, the angiotensin converting enzyme 2, is also located in a lipid raft. So that is very convenient for this SARS-CoV-2, the virus, because it has this spike protein, and the spike protein binds to the ACE2, and the ACE2 is located in the lipid raft itself. And then once it has come and got attached to the surface of the cell by means of this lipid raft, then the viral envelope, the envelope of the virus, will undergo enzymatic activation, and that enzymatic activation also happens within the lipid raft itself. And then the organism gets endocytosed, and it is within an endosome, which is also raft dependent endosome formation. And then after internalization, it will undergo intracellular trafficking. As you can see, it will go to the lysosome, and from the endosome, it will fuse with mature lysosomes, and it will release the viral RNA genome into the host cytoplasm. And then if it has to transmit from one cell to another, there also it uses the lipid rafts for the organism to go from one cell to another, showing that the SARS-CoV-2 is actually exploiting the lipid rafts for its entire life cycle. There is the initial binding, the activation, and the internalization, then the cell transmission. All of them will require intact host lipid rafts to proceed. So if it's not only the SARS-CoV-2, HIV, the human immunodeficiency virus also, they had seen that, it is by means of the lipid, it also comes and binds to the lipid raft through, the cytokine, the CCR5 and the CCR5 and a co-receptor. And finally, there'll be clustering of the CD41, I think it is Gp41, the glycoprotein 41 on the lipid rafts, and that is the one which allows a human immunodeficiency virus to actually bind to the cell surface and cause the disease. This clustering of these receptors becomes important, and that happens within these lipid rafts. So you can see how microorganisms or pathogens like the more recent SARS-CoV-2. It's not only the SARS-CoV-2, even the other the mother SARS-CoV, which was actually then mutated. The spike protein was mutated to give the SARS-CoV-2. That also uses the lipid rafts for its attachment and for its entire, or not entire life cycle, at least for cellular transmission. These are used. So why do we have to look at this at all? Let's look at another one, and as I said, my favorite organism, which is your H. pylori. Now the H. pylori also manipulates the host cholesterol, which is enriched in the lipid rafts. Actually, the cholesterol in the lipid raft is like a glue there. It just holds everything together. So the H. pylori is actually exploiting or manipulating the host cholesterol for infection. The H. pylori produces two toxins, as I told you. One is the VAC-A and the CAG-A. And it's a CAG-A which is injected into the cell later on. So first it is a VAC-A toxin which is secreted by H. pylori, and the toxin, not the organism, but the toxin will go and bind to these lipid rafts, and they are all microdomains which are rich in cholesterol. And then you will find they will all come together, aggregate like this. These lipid rafts will aggregate, or coalesce as they call them. They will aggregate, and the clustering of these rafts will initiate, as you can see in the next slide, will initiate the secretion of CAG-A into the cell. And I told you Helicobacter, there will be invagination of the bacteria, and the CAG-A can be secreted into the cell, because H. pylori doesn't invade a cell, a gastric epithelial cell. It always sits on the surface and only injects its toxin into the cell, and it is the CAG-A that is causing all the all the symptoms that you see during the pylori infection. And And as you can see here, the raft associated, now these are all membrane, the membrane which is associated with the raft which is invaginated there. And these raft associated membranes will extend to form autophagosomes. And these are compartments for the H. pylori to survive intracellularly, but it doesn't actually, not in all cases the H. pylori enters into the cell, but it even if it enters, it is going to be enclosed in a phagosome so that it is not released intracellular. Here you can see that the foothold for H. pylori is also a BC one, that is one with all these lipids, specifically the cholesterol. So when we look at all these, what do we think? Why am I connecting SARS-CoV-2 lipid raft interaction and the H. pylori lipid raft interaction? I'll come to that later, or in the next slides for that, but I just want to inform you about it. So like the importance here is the lipid rafts are very important. Only if they are intact, then that the organism will be able to bind, and it can go through its life cycle. But then it also means that if you can disrupt the lipid rafts, then the organism will not be able to survive on the cell surface and therefore cause an infection. So more recently people have been thinking about this, where they have used cyclodextrins as well, which can actually break the cholesterol there, and also statins which can reduce cholesterol levels, because statins, we know, inhibit the first step, that is the reductase step in cholesterol synthesis, and reduce cholesterol levels. And that way the raft, the stability of the raft is going to be disturbed, and the organism will not be able to colonize there. So, some people say even for the COVID-19, statins will be helpful, but I don't know whether these are all hypothesis and theory that we are looking at, and with any kind of clinical trials have been done is not known. So now, why did I, and also, when you look at the proposed mechanism of gastric carcinogenesis, the obvious step is binding here. It is binding to a cell surface carbohydrate, and it can also bind to a cell surface, lipid raft, and inject this CAG, and all these signaling mechanisms, which will finally lead to uncontrolled cell proliferation, as we see here. That's for H. pylori. But then what is this relationship between the H. pylori and the, what is that called, your SARS-CoV-2 or the COVID-19? Now, more recent, so after, we have been doing a lot of, not we, I mean, scientists all over the world, because as I said, almost half the world's population is infected with H. pylori, and therefore people have been doing a lot of research to actually eradicate this H. pylori, mainly in developing countries. So even after doing so much of aggressive research and trying to convincingly demonstrate the pathological outcomes of H. pylori and the necessity to eradicate the H. pylori, a new controversy has emerged now. Now, more recently in 2020, late 2020 and early 2021, there have been reports from some of these developing countries that, there are two kinds of reports, right, which are controversial. The first one says that if a person is infected with SARS-CoV-2 and has COVID-19, and if that person also has an H. pylori infection, then he is not going, the severity of the disease is going to be much less, because the H. pylori in some way or the other will protect the host from the infection, the severe infection caused by SARS-CoV-2. So therefore, don't ever think of doing a widespread eradication of H. pylori. Let it be there, because if it is going to be there, then the severity of or no, not even severity. Now you know that patients with COVID-19, they also have gastrointestinal symptoms like severe stomach pain and diarrhea, etc. So all those symptoms will not be seen if the the COVID-19 and the H. pylori are going to live together there. So that is one theory that has been put forward. It's not just a theory. In a few, say something like 100 patients with H. pylori, COVID-19 with H. pylori infection, COVID-19 without H. pylori infection, they have been able to show. Then there is another school of thought, another kind of report that I saw recently, and that says that if you have H. pylori infection, then the severity of COVID-19 will be much greater, because H. pylori is supposed to upregulate the expression of A2, the angiotensin converting enzyme 2, and that is the receptor for the SARS-CoV-2. Therefore, if there is a H. pylori infection in a person, then ACE2 levels are going to be in, and that is like inviting the SARS-CoV-2 to bind to the receptor, and the severity of COVID-19 is going to be much bigger. Now, which one of these are we to believe? And I just showed this slide just to show that after doing so many years of research, and not only, I'm not talking about me, but several others who have done. You say don't eradicate H. pylori, don't think of ways, because here we have looked at one point here, a drug which can inhibit this step, a drug which can inhibit this step, and then finding out or identifying such drugs are not going to be of any use in this scenario where you have this COVID-19 pandemic. Now, why do we say that if there is H. infection, COVID-19, it is going to protect against COVID-19 or the SARS-CoV-2 infection extent? Because there have been reports like when, again, this is like a competitive inhibition maybe, because H. pylori is also located in these wraps, the lipid wraps. So that is why these lipid wraps are very important in entry points, and if we can understand these entry points, and we can think of ways in which you can actually disturb them and prevent the organism from causing the disease. So there are reports like Campylobacter toxin, when it binds to its receptor in which is located in lipid wraps, that prevents the vacuum of H. pylori and the caddy of H. pylori. Initially, it was a vacuum which comes and binds to the lipid wrap, so it prevents the vacuum from binding. So there is a kind of a competition between these toxins, between these organisms for site of lipid raft. So maybe there's something like that here which we do not know. It's just a hypothesis that COVID-19 SARS-CoV-2 in some way is preventing or is being prevented or is being protected against SARS-CoV-2 binding by infection when the patient is infected with H. pylori. So a lot of controversies and whatever it may be, is that initial entry which is very important, and if that can be identified, then you can design therapeutic strategies, you can design drugs to block that initial entry point, but that is not going to be easy. and might disappear. So I would like to stop with this. Did I take too long? Yes, someone, anybody, you can unmute now and
Participants, if you have
no.
any doubts, you can ask, ma'am.
Have all the participants finished their oral presentations and poster presentations?
Dear
Yes,
participants,
ma'am. Yes,
if
ma'am.
you want to ask some questions, you can ask.
Probably they're tired after their own oral presentations.
Ma'am,
Yeah, the natural one, Muttu, I think, right?
Yes, ma'am.
Muttu
Yes,
has a question, and that is there any natural inhibitors of additions? Yeah, I mean, most of the time, the kind of research that we do in biochemistry is to identify natural compounds which can be used to, inhibit these additions. So we have seen some flavonoids and some terpenes, etc., which do inhibit the initial adherence. But as I said, these are all done in vitro, and whether that can be extrapolated to in vivo conditions is something that is not known. So if you can actually identify a natural inhibitor, it would be good, but there are several reports where they say, especially with this in the YouTube, they give you so many different natural products that can be used for the treatment of any disease for that matter. And but I don't know how far these will work. But if you can really identify and if you can really prove that these natural products can inhibit, then it'll be good. But I don't think anybody has done it so far. Okay, yeah, Muttu.
Any other questions, participants?
I'm thankful that I was able to complete this because, this thing kept saying that your internet connectivity is poor and, do some troubleshooting, but, it really didn't interfere with whatever I had to say. Any other questions? Probably the participants also have a problem with their connectivity.
Yes, ma'am. Yes.
That's because are they all from Chennai or from different
No, ma'am,
places?
from different places.
Okay, so
All over India, ma'am.
All over India.
Some people are West Bengal, Assam, Delhi, and all.
Okay, so that definitely I'm sure they're finding it difficult.
Yes,
and, but maybe it's not raining there.
Yes.
Okay.
Ma'am.
Yeah. Yes, ma'am.
Yes. Ma'am, shall we wind up the session, ma'am?
Yes, ma'am. Sure, ma'am. I'm sure everyone is hungry. And anyway, thank you for the opportunity.
Thank you so much, ma'am. Thank you so much, ma'am. It's
thank you, ma'am. Ma'am, can you ask ma'am to on the camera? You can take a photo, ma'am. You can take a photo, ma'am.
Switch on your camera, ma'am.
Yeah. Some light is on, then I don't know.
Yes, ma'am. I'll take a photo, ma'am. 1 second.
Yeah, sure. But it's all, I don't know, maybe you see a better figure there, or what I'm seeing is just some.
Yes, ma'am. Yes, ma'am. Same thing only. In the mirror, the light is
very dark.
Yes.
Okay, anyway.
Actually,
okay.
Indu, ma'am, is the photograph clear, ma'am?
No, ma'am, not ma'am. Ma'am, the light is coming, ma'am, above.
Yeah, and the light off panava, Elena.
Yes,
ma'am, you
please,
try.
ma'am.
Yeah, I'll do that. Actually, I tried it, but when I did that, there were like some shadows. There were shadows, and therefore I couldn't.
Ma'am, it's okay, ma'am. Now it's okay, ma'am. Now is it okay, ma'am?
Ma'am, now it's clear, ma'am.
இப்பதான் நான் உள்ளே
Now it's okay, ma'am.
Yes,
Okay,
ma'am.
Yeah, this is okay, ma'am. This is okay, ma'am.
Yes.
maybe on the light at the back is not.
Yes.
This is on the side, but idile some shadow madri something is shaky there, right?
Oh,
1 second.
ma'am, have you taken the screenshots, ma'am?
Yes,
You,
ma'am. Yes, ma'am. Yes, ma'am. Thank you, ma'am.
Okay, ma'am. Personally, I feel so happy after because I hear you after so many years, ma'am. It's really privilege to have you on our conference, ma'am. Thank you so
You've
much,
been
ma'am.
in which batch did you do your
Ma'am, 2006 to 8, ma'am.
6 to 8? That's a long time back.
Yes, ma'am. Yes, ma'am. And really, you were an inspiration to me, and I used to think you often, ma'am.
Anyway, thank you, ma.
Thank you, ma'am.
2006 la irundha, if you can remember me, that's good. I'm happy.
Ma'am, no one can ever forget you, ma'am. Kirtana, you can proceed, ma'am.
Yes, ma'am. Thank you, ma'am. You have made an everlasting impression on everyone. It was obvious that you are extremely knowledgeable in your field, and a link of this knowledge to a great presenting style was impactful. I want to thank you once again for providing us such an informative speech, ma'am. Thank you so much, ma'am.
Thank you, ma. But I was under a lot of attention the past 1 or 2 days because of this, the internet connectivity and also the, the rains in the morning. It was raining here, and I'm thankful that I was able to complete this. Thank you, ma. So can we leave now, or you have some sessions in the afternoon, isn't
Yes,
it?
ma'am. One more session is there, ma'am.
Okay, right.
Thank you, ma'am. Thank you for your presentation.